All right, today we're very excited to have Adam Siepel speaking on phyloseminar. This is the second talk in the series in evolutionary genomics. The next talk in this series will be Jason Steich speaking on June 29th at noon PST. So we do encourage questions during the talk, and if you have one, you can do one of two things. First, you can just type your question in the message box, or you can just type in there that you have a question, and I'll turn on your microphone. If you do decide to do that, though, make sure that your audio is debugged beforehand. So, as I mentioned, today's speaker is Adam Siepel. I was most familiar with his work on phylo-HMMs with David Hausler and others, but it was pretty cool looking through his list of publications. So his first publication is a computer program for screening HIV for recombinant, okay, with Betty Corder. That's pretty cool, back in 1995. And he got interested in gene order phylogeny, okay. And then I saw this paper, a simplified hill slope erosion model with vegetation elements, etc., etc. Anyway, that's pretty cool. I should let him get on with his talk, but he's done all sorts of really interesting stuff, and we're really happy to have him speak to us today about Bayesian inference of ancient human demography from individual human genome sequences. All right, thanks.
Great, thanks, Eric. So let me just get a sense for how many people I'm talking to here. I only see like 4 names, including me. Is that the complete list, or is that a sub-list?
Well, that's the complete list of remotely connected people. We got Connie here too.
Okay, all right. So it's a very small group. So given that the group is so small, I encourage people to feel free to interrupt with questions. Okay, thank you. I was hearing some feedback, but it's gone now. So you talked about the diversity of projects we've worked on. Actually, that hill slope erosion model was my undergraduate thesis at Cornell, which got published 7 years later. We haven't done any work on soil erosion recently, but we have been moving into population genetics much more heavily than we had been in the past. And this has been really exciting to us because it's quite a new area. We've been doing it in a way that allows us to use some of the things we've learned about phylogenetics and molecular evolution over the years, but nevertheless, it's very new data types, and the type of signal in the data is much different from what we're used to working with in a comparative genomics or phylogenetics setting, and this has taken a lot of adjustment and a lot of thought. So I'm going to talk about, I'm basically giving you a slightly padded out version of the talk I gave last week at Cold Spring Harbor. And I didn't have to pad it too much because my talk was a bit too long to begin with. So this is very new work. The talk will come in 2 parts. The first part is nearly published, although not quite, and the second part is very new, preliminary work, and you'll see that in the results. There's a lot we're unsure of with our results so far. So let me launch ahead. First, I'll just mention on my cover slide here, I guess I can do the pointer this way, right? So this work is led by Ilan Grunau and Leo Arbiza. I see the pointer moving slowly across the screen. 2 postdocs in the group. And then we've had also contributions from a number of others. It's really been a team effort, including Armand Aksoy, a very talented rotation student, Charles Danko, another postdoc, Melissa Hubis, a staff member in the group, and Brad Gulko, a grad student. Oh, now I lost my focus. Okay. Okay, so what we're doing in this project is analyzing the complete genomes of individual humans. As many of you know, I'm sure there's been a lot of interest in the past few years in sequencing individual humans, sort of culminating in the 1,000 Genomes project, where a large number of individuals were sequenced at low coverage. There's some other very rich recent data sets coming out, including a data set from the company Complete Genomics consisting of 60 whole genome sequences for individual humans, high coverage genome sequences. There's a lot of data in this area, but I think it's fair to say that that most of the interest recently has been sort of on technical feasibility of genome sequencing, particularly using next-gen technologies, and on potential biomedical applications of whole genome sequencing, implications for personalized genomics, and so on. We've been interested in taking a more explicitly evolutionary perspective on these data. And so that's what I'm going to talk about today. The talk will have two parts, and those two parts really address two different aspects of interpreting individual genome sequence data, one concerned primarily with demography and one concerned with natural selection. So in this first project, we were interested in using these newly available genome sequences to gain insights into the very early divergence events between human populations, most likely in Eastern or possibly Southern Africa. So the events that occurred on the order of 100,000 years ago, by which the earliest splits occurred between major human population groups that are still existent today. And we were interested in particular in making use of data sets that, two genome sequences that became available about a year ago. One of them describing a San Bushman hunter-gatherer individual from the Kalahari Desert in Namibia, and one of them describing Archbishop Tutu, a representative of Bantu populations in southern Africa. South Africa. And this Bushman genome, which I'll refer to as the San genome, was of particular interest because it's believed that these people, based on mitochondrial DNA evidence, Y chromosomal evidence, and some autosomal evidence, they appear to be one of the most divergent groups on Earth today, and so they should be particularly informative about these very early events. So in analyzing these data, we relied on a fairly long line of literature going back to the mid-'80s in molecular evolution, phylogenetics, and population genetics, by which people have tried to figure out ways of basically disentangling two contributions to genetic divergence. So a contribution corresponding to the time since populations or species have diverged, denoted tau in this figure, and the time that it takes for individuals within ancestral populations to find their most recent common ancestors, so-called coalescence time. I'll refer to this as the ancestral coalescence time. And if you have a single locus in a genome, as you have when you analyze a Y chromosome, the non-recombining portion of the Y chromosome, or a mitochondrial genome, you basically have no information that allows you to disentangle these two components. But when you have multiple loci across the genome, you can use variation in the genealogies at these different loci to separate these two components. And without going into a lot of detail, the basic idea is to use variation either in divergence times or in tree topologies along the genome to characterize the ancestral coalescence process, to gain information about ancestral effective population sizes. And then once you've characterized that ancestral coalescent process, you can explain sort of the remainder of the divergence in terms of divergence between populations. Obviously, this is sort of easier said than done. The devil's in the details in making this sort of method work. There are lots of confounding factors that have to be considered in separating these two components. So first of all, there's uncertainty in distance estimation, uncertainty in tree estimation. There are factors such as mutation rate among loci that can contribute variance that, unless it is properly modeled, can appear as a contribution to the ancestral effective population size. And so you need a fairly sophisticated model and fairly carefully curated data sets in order to accomplish this sort of analysis with genomic data. Um, there's been quite a lot of interest through the last decade or so on improving statistical methods for separating these two contributions to genetic divergence. And from our point of view, the sort of Cadillac of the methods that have been developed in the last 10 years or so is this method called MCMCcoal, developed by Ranala and Yang in 2003, actually building on several papers by Ziheng Yang in the late 90s and early 2000s, and then subsequently refined by Burgess and Yang in 2008. And MCMCcoal is a fully Bayesian method that basically works with three types of objects. So you have a set of loci across the genome. They're selected so that they're short enough that you can assume negligible amounts of recombination within loci. So you can assume essentially a single genealogy per locus. They're spaced far enough apart along the genome that you can assume they're essentially independent. You can assume free recombination between loci. And then for each one of these loci, you have a multiple alignment representing each of the individuals in your sample. In addition, you have, as I'm showing on the right here, you have a population phylogeny whose topology you can assume is known. And that topology is characterized by a set of pre-parameters describing the effective population sizes of all ancestral populations and the divergence times associated with each of the bifurcations in the phylogeny. So we'll refer to those as theta parameters and tau parameters. Typically, they're expressed in units of mutations, expected numbers of mutations per site, and then later on, they can be re-expressed in terms of numbers of individuals or in terms of numbers of years by making use of some external calibration, either a mutation rate calibration or a calibration with a particular divergence time of interest. In our study, we'll use the human-chimpanzee divergence time as our calibration. So MCMCcoal makes use of these two objects, the alignments per locus and the population phylogeny, but then it makes use of a third type of variable, which is actually a latent variable. It's not fixed throughout the analysis. It's treated as a random variable that's sampled by Markov chain Monte Carlo methods. And these are individual genealogies per locus. So the method essentially samples over possible genealogies at each of the loci of interest, effectively integrating them out as a nuisance variable in the analysis. And it attempts to get at a, oops, I lost my mouse again. It attempts to get at an approximate posterior distribution for these free parameters of interest, the theta and the tau parameters, given the data by Markov chain Monte Carlo methods. And it does this, in doing this, it exploits the fact that when you introduce the genealogies when you augment the variables with an explicit representation of the genealogies, the likelihood calculations become fairly straightforward. So the probability of each genealogy given the model parameters can be computed using relatively straightforward adaptations of the standard coalescent, and the probability of each alignment given a genealogy can be computed using standard methods, essentially using Felsenstein's pruning algorithm, with whatever nucleotide substitution model you choose to use. So the likelihood calculation is straightforward. The challenge is, as always with these MCMC problems, is in developing a Markov chain Monte Carlo sampling method that effectively explores the space of possible models, that mixes well, and that has good convergence properties. Okay. So our goal, somewhat naively, was to apply MCMCcoal or an MCMCcoal-like method to the available individual genome sequences. And we did this, we launched this project more than a year and a half ago, and at the time there were quite a few fewer genomes available than there are now. So we settled on one genome per population with 6 different populations. So we had a Korean individual, a Han Chinese individual, a European American, a Yoruban, West African individual, a Bantu, the Archbishop Tutu, the Bantu representative from South Africa, and the San individual. And we used the chimpanzee reference genome as an outgroup, as I mentioned. And we used the following tree topology shown here, which was in general not really in question and well supported by preliminary analyses just looking at overall levels of divergence between genomes. So we have the two East Asian genomes most closely related, the European genome being the next closest genome, the San genome being the earliest branching of the populations under study. And then we had some difficulty in resolving this relationship between the Yoruban and the Bantu genome. And we're never able to resolve this in a completely confident way. So the way we ended up addressing the relationship between these two genomes was to analyze them both separately. So we would include either the Yoruban or the Bantu in our analysis and perform the whole analysis using a tree like the one shown here. And then we could cross-check the two analyses. We could cross-check our results based on the Yoruban and our results based on the Bantu. And that was actually sort of a nice control in the analysis that allowed us to study its robustness to the inclusion of a particular group. And this is a key group. We call this the African in-group. It's probably the most important of the genomes, in addition to the San genome itself, for dating the San divergence event. So we were particularly interested in this study in two early divergence times, the divergence time of the San, which we refer to as the San divergence, and the divergence time of the Eurasian individuals, the Korean, Han Chinese, and Europeans, relative to the African in-group. We refer to this as the African-Eurasian divergence, or AE divergence. We also looked at some of the other divergence events, but as it turned out, we had much less power to estimate those accurately, basically because the methods that we're using are driven by mutations. So the signal for divergence times comes from the accumulation of divergence along genealogies, and there are very, very few mutations separating these genomes that are more closely related. So we focused our attention on these more ancient events. And then I should say we used a calibration of 6.5 million years for the human chimp divergence. This is the average genomic divergence, so not the speciation time, but the average divergence time across loci between n individual human and n individual chimp. But we also considered a range of a fairly inclusive range of possible divergence times from 5.6 to 7.6 million years. And all of our estimates end up just scaling linearly with that assumed calibration time. Okay, so in actually making this work, we faced a whole series of difficult challenges, and I'll talk briefly about some of them and about the solutions that we came up with for addressing these challenges. In this first slide, I'll just sort of lay them out, and then I'll show you a few more slides where I say a few more words about each of the solutions we used. The first problem, which turned out to be really a crippling problem, and it, for some time looked as though it might force us to abandon the entire project, was getting these genome sequences, these available genome sequences, in a form where we could reasonably compare them. So I don't know if you can tell from the slide, but I list the technology used to sequence each of these genomes, and there were 4 different technologies used to sequence the 6 different genomes that we analyzed. They also had very different coverage levels, ranging from about 7x to more than 40x. Different read lengths. Some of them were paired end reads, some of them weren't. So we had very, very heterogeneous data sets. We initially tried using the SNP calls that were publicly released along with the papers that describe these these individual genome sequences. And we found we were able to get basically nothing out of the publicly released SNP calls. They were not only based on very different data sets, but also based on different alignment methods, different bioinformatic pipelines for SNP calling, and so on. So what we ended up doing was spending a lot of time going back to the drawing board, building our own genotype inference pipeline, involving realigning all of the raw reads that had been released by these genome sequence projects, developing our own Bayesian genotype inference method, recalibrating the quality scores that had been produced by these different sequencing technologies, and so on. And I won't say too much about this. There are a lot of not so interesting details involved, but I'll show you a few slides highlighting what we were able to accomplish with this uniform genotype inference pipeline. The second major challenge was that the available methods for doing this type of inference, including MCMCcoal, assume no gene flow between populations after their divergence. And that's obviously a major problem with human populations, for which there's lots of evidence for gene flow subsequent to population divergence events. And so we ended up, in order to address this, making some fairly major modifications of the MCMCcoal source code. In fact, we modified it enough that we felt justified in renaming our modified version of the software. So this was work done by Ilan Cornell in my group. He calls the modified program GFOX for Generalized Phylogenetic Coalescence Sampler. And as I'll show you in a few slides, GFOX has a general method for sampling genealogies that are allowed to cross the boundaries between populations in this population phylogeny, and it can estimate parameters describing rates of migration between populations. We were able to show, using simulation methods, that we could accurately recover the parameters of interest, even in the presence of gene flow, by adequately modeling the gene flow process in this way. The third problem we faced in making this work was the problem of phasing with the data. So this is sort of a more technical issue, but still an important one. The data that we were working with was all unphased diploid genotype data. MCMCcoal basically assumes haploid samples. We could have simply run a phasing algorithm on these genomes and tried to pre-phase them, but we were quite worried about the effects of phasing error on our inferences, and we think phasing error could be fairly non-negligible with these samples, because some of the samples of interest have good reference panels for phasing, like the Yorubans and the European Americans, but others, like the San and the Bantu, really don't. So we expect we might have considerable phasing error in those samples and very heterogeneous amounts of phasing error between samples that could produce serious biases with our analyses. So instead, we took a different approach. We analytically integrated over all possible phasings in the inference. And this turned out to be a relatively straightforward algorithmic solution to the problem, but one that, as far as I know, hasn't been done before. The solution that we used is not highly general, but it works well for small sample sizes, so small numbers of individuals at the leaves of the genealogies of interest. In our case, because our number of samples is small, and the number of heterozygotes per site is relatively small, we end up only increasing our overall computational cost by about 10% by integrating overall possible phases rather than fixing the phase and working with haploid samples. And we can show in simulations that we're able to recover estimates that are essentially as accurate as what you would recover by conditioning on the correct phase in this setting. So we were quite happy with that. And it allows us to avoid any pre-processing for phase and any sensitivity to phasing error. So this is just a quick slide showing our genotyping pipeline. We start with the raw reads, as I mentioned. We line them with BWA. We use the GATK tool from the Broad Institute to recalibrate the quality scores, use SAM tools to create a so-called pile-up representation showing all of the observed bases at each position in the reference genome. And then we developed our own method, which I'll say a word about in a minute, called BSNP, for inferring the genotype based on those aligned reads at each position. BSNP is similar in spirit to SOAP-SNP and some of the other Bayesian genotype callers that are out there. The main difference is that we avoid conditioning on the reference allele at each position in the genome. So we go out of our way to avoid using the reference allele or anything from HapMap or dbSNP as a prior in our genotype inferences, because our expectation is that those priors, especially with the lower coverage genomes, could have quite different effects on different genomes, and they would essentially be pulling each of the genomes toward the reference allele or toward the dbSNP allele in a way that depended on the amount of data in each genome, and could give us a sort of a biased representation of the genotypes in these different genomes. We also then use a large number of samples, of filters. I'm going to get rid of this, get rid of that pointer. We also use a large number of filters, heavily filter the data to eliminate regions of lower read depth, regions that are difficult to align because they contain recent transposable elements or simple repeats. We filter the data heavily to ensure that we have high quality alignments with the chimpanzee genome, making use of SNP information. We avoid segmental duplications, and then we avoid all regions in the genome that are likely to be under selection, including protein coding genes, non-coding RNAs, conserved non-coding elements identified by FASTCons, and so on. Because we have so much data, we're able to filter it quite heavily and work with a subset that we think should be highly depleted for both data quality errors and the effects of natural selection. Just a few words on this genotype inference problem. I don't want to say too much about this, but people are probably familiar with the problem. Typically, you have a reference genome, you align reads from a bunch of individuals to the reference genome, and then you scan across the reference genome position by position, and at each position you make a decision about, you make a call about what the genotype is, whether it's homozygous or heterozygous, based on the pattern of reads that are aligned at that position, allowing for sequencing error, missing data, taking into consideration useful covariates such as quality scores or mapping scores, and so on. As I mentioned, the difference between our method and most previous methods is we avoid making use of the reference allele that's observed at each position in deciding which genotype is most likely. So we're basically just using the reference genome as a scaffold for alignment, as something that allows us to sort of hang the reads observed from the different individuals to position them in a consistent manner. But then we ignore the actual identity of those reference bases in subsequent analyses. We use a Bayesian method based on a graphical model that looks something like this. The G variable at the top here represents the unknown diploid genotype, so there are 10 possible diploid genotypes, allowing for both homozygous and heterozygous genotypes. We assume that the two haploid alleles associated with a diploid genotype are sampled uniformly, each with 50% probability, and then based on those unobserved true haploid alleles, we observe in the data essentially a noisy representation of the true base, indicated by the X variables in this picture. And how noisy a representation we observe, we can measure essentially by looking at the quality scores and the mapping scores at each position. So it's not difficult to write down a likelihood function and to come up with a little algorithm that allows you to infer a distribution at each position over all of the possible genotypes. We actually started out in this work working fully probabilistically with the genotype. So we worked with full posterior distributions over genotypes at every position. In the end, we found that even with relatively low coverage data, those posterior distributions were highly peaked. And so we ended up just working with the maximum a posteriori genotype call at each position because it simplified the analysis considerably. We did a lot of work validating these genotype calls. I won't say too much about it, but here's a figure from our supplemental material for our paper showing that we're able to perform essentially as well as, and in this case slightly better than 1,000 genomes calls or calls based on the MAC genotype caller. This is based on a validation study of one of the European genomes sequenced by the 1,000 Genomes project to high coverage. We also looked at the Venter genome, comparing it with array-based genotype data to assess accuracy, and similarly showed that we were able to call genotypes quite accurately. What we were particularly interested in, as well as accuracy, though, was the uniformity of the calls across the different genomes in the sample. So this is maybe a little bit too complicated of a figure, but if you concentrate on the two rightmost bars for these different samples, those represent rates at which heterozygotes are called across the genome. So you can think of them as just estimates of per site heterozygosity. And the blue bars, so the second from the right, the light blue bars, represent the published heterozygosity values. The final bars, the sort of pinkish ones, represent our estimates of heterozygosity based on our genotype calls. And you'll see that the blue ones, the published ones, are quite wild. So there's no clear pattern, no clear consistency between the genomes representing the same populations. Whereas our genotype calls are quite a bit more consistent. The two European genomes have similar levels of heterozygosity as would be expected. The two East Asians also have similar levels of heterozygosity as would be expected. And the three Africans have roughly similar levels and considerably higher levels of heterozygosity than the Europeans and the East Asians. So this analysis and a number of other sort of simple summary statistic-based analyses gave us some confidence that we were doing a reasonable job of putting all of these genomes on the same footing and making it possible to do a population genomic analysis of these data. So this is just a picture showing what the modifications to the sampler have to accomplish. So in this setting, you essentially have a population phylogeny. We sometimes refer to this as a tube tree. You can think of it as a tube within which genealogies are sampled as you go across the genome. If you ignore migration, then the genealogies that you sample have to be completely contained within that tube tree. But if you allow for migration, then you permit occasional sampling of genealogies that cross boundaries between these populations. The way we organize this is we don't allow migrations to occur absolutely anywhere in an uncontrolled manner. The user has to establish particular migration bands between populations within which migration is allowed. And we end up running our analysis multiple times with multiple different migration bands and comparing results. We found that if you add too many migration bands at once to the analysis, you just can't get the Markov chain to converge. The problem just becomes too unconstrained, and you run into serious problems with mixing and convergence of the MCMC algorithm. Well, I don't want to get ahead of myself, but we end up seeing evidence for migration between the African populations, but much weaker evidence for migration between the other populations over the timescales that we're interested in. Okay, yeah, so just a few sort of technical points about this. Going back to this decomposition of the likelihood calculation used by MCMCcoal into what we call a genealogy prior and a data likelihood. The genealogy prior is the main part that needs to be modified, when you accommodate migration. So this is the probability of each genealogy given the model parameters. When you introduce migration, you now have to be able to compute the probability of a particular genealogy that may or may not cross population boundaries, given not only your split times ancestral effective population sizes, but also your migration rates within the allowable migration bands. The locus data likelihood, on the other hand, isn't affected at all by the introduction of migration. Remember, the locus data likelihood is just the probability of a particular alignment given a fixed genealogy, so essentially given a tree with a set of known branch lengths. That once you fix the genealogy, you don't care whether or not that genealogy arose via migration in calculating the probability of the data. So this portion of the likelihood function can still be calculated using Felsenstein's algorithm with whatever substitution model you like in the same way as it could in the standard MCMCcoal method. So, excuse me. When all is said and done, we end up with a model that looks something like this. We have a phylogeny, a general phylogeny. Each of the branches of that phylogeny represents a population, either a present day or an ancestral population. There's an effective size scaled in units of mutations, so a theta parameter associated with each of those populations. And then for each of the divergence events in this population, each of the splits in this population phylogeny, there's an additional parameter representing the time at which that split occurred. And then there's some user-defined collection of migration bands between populations, each of which has its own free parameter describing the rate at which migration occurs. And then the method attempts to estimate all of these model parameters, the theta parameters, the tau parameters, and the migration parameters, which we call M, using the Markov chain Monte Carlo sampling methods. Just a few quick notes about what has to be done to make this work. Here's a sketch of the MCMCcoal algorithm published by Bernala and Yang in 2003. It basically goes back and forth between steps that update the locus-specific genealogies, such as steps 1 and 2 here, and steps that update the population level parameters, steps 3 and 4 here. And then it has a step that does some global scaling of all of the theta and tau parameters that is designed to cause the sampler to mix better for reasons that I won't go into. Step 1 is a simple stochastic resampling of the individual coalescence times within each genealogy. Step 2 is a genealogy topology resampling step, and that's done by a subtree and subtree pruning and regrafting step. So what do you have to do when you accommodate migration? Well, the way we approached this problem was closely related to what's been done in some other work, most notably the IM and IMA programs by Jody Hay and his group. When we do the regrafting step, so after a subtree pruning step, when we regraft a subtree onto the remainder of the genealogy, we allow lineages to pass through migration bands, through the designated migration bands. That's actually the only way in which migrations can be introduced. New migration events can be introduced during the sampling. That turns out to be adequate. We need a new step, however, to deal with the timing of the migration. So similarly to the way the coalescence timing events are resampled in the ordinary MCMCcoal algorithm, we have to resample the migration, the migration times. And then we also need to add a step for a new global parameter describing each of the migration rates, one per migration band. And then some of these scaling issues that I glossed over also need to be modified to deal with times of migrations as well. There are some complications leading to conflicts. Essentially, the introduction of these migration events between populations creates dependencies between what could previously be described as independent events happening in these different populations, and you need the code needs to have some awareness of these possible conflicts and to resolve them. Okay, I won't spend any more time on that, but if anyone is interested, I can talk more about some of the details in making this work. It's actually fairly technically challenging to get the Markov chain Monte Carlo method to work with migration. Just a quick note about the phasing integration. So we basically assume there's some latent variable representing the phase of all of these diploid alleles, and we integrate overall possible values of this random variable, of this matrix describing phase. If you assume a uniform prior across all of the positions, which is fairly reasonable since phasing is essentially an arbitrary decision, and conditional independence of the sites given the phasing assignment, then you can basically move this sum over possible phases inside of the product over sites. And then you end up having to sum over phase separately at each individual position within an alignment. The number of terms in that sum is exponential, as it turns out, only in the number of heterozygous genotypes per position. And in our case, that number tends to be quite small. We've only got, at most, 5 genomes in our analysis. Typically, they're not all heterozygotes at any particular site. So we can sum over all possible arrangements of those heterozygous genotypes at the leaves of the genealogy relatively cheaply, with a small cost, small overhead in total computation. And the likelihood calculation then becomes just a sort of average over all of these possible phasings at each individual site. So it's not a particularly sophisticated idea, but it ends up eliminating the problem of phase and working quite well in practice. I think I'll skip over this slide. There are a number of steps we go through to improve the efficiency of the method. Efficiency is quite important in this setting. We're working with tens of thousands of loci in a genome-wide analysis, and we have to get these sampling methods to operate very efficiently in order to be able to produce results. So we went through. Yeah.
Just a quick question about the phasing. I thought that on the previous slide you said that you picked the phase. I think I'm confusing two separate things. You said that with the phasing that it was quite peaked.
The genotype, the particular genotype at each position is peaked, and we fix that. So at every position, you might have, you've got 10 possible genotypes, an AA homozygote all the way through, so there are 4 possible homozygous genotypes, right? And then 6 possible heterozygous genotypes, an AG, an AT, an AC, and so on. Of those 10 possibilities at each particular site, we pick one based on the BSNP analysis. The problem that we're facing here is, as you're looking across a particular locus, our loci are 1,000 bases in length, you may have heterozygous genotypes at different positions along that locus, and you don't know how those heterozygous genotypes align with one another. So if you have an AG at one position and a CT at another position, you don't know if the A goes with the C or the A goes with the T. And that's the phasing problem, and that's the part we integrate out.
Okay. Thanks. Sorry about that.
Sorry about that. Yeah, I probably need a picture with the phasing, illustrating the phasing a little bit better. Okay, so I think I'm starting to run short on time, so I'll go quickly through our simulation experiments. The basic goal here was to show that aside from issues of making sure the software actually worked and converged and operated properly, didn't have bugs, we needed to show that we had enough information in the data at the timescales of interest to be able to extract our parameters of interest. So we simulated data sets similar to what we expect the correct answer to be for human populations. We worked with similar numbers of loci to what we have in our real data. Actually, somewhat fewer loci. We used 20,000 loci in the simulations. We worked with a 5-population phylogeny like the one that we're using for inference, and considered a range of possible parameter values sort of within the range of plausibility. And then did inference both with and without migration and with and without known phasing. And the bottom line from these results was that, as I'm showing on the left here I'm showing both the correct, the true values and the estimated values for our two divergence times of interest. The first one, tau KHEX, represents the African Eurasian divergence time. The second one, tau KHEXS, represents the SON divergence time. I'm showing 3 possible true values indicated by dotted lines, and then a graph indicating the range of possible, sort of a box plot indicating the range of inferred values over a set of different experiments. And you can see that we're able, with reasonable accuracy, to recover the correct values of these parameters. And when we do this by integrating over all possible phasings, we do essentially as well as when we know the correct phasing in a simulation setting. You can see also that the estimates are considerably more accurate for the deeper divergence time, the SON divergence time, than they are for the more recent African Eurasian divergence time. And when the African Eurasian divergence time gets quite recent, so you see that true value of 0.3 times, that's times 10 to the minus 4 mutations per site. Don't worry about the units too much. But when we make that divergence time very, very recent, we do start to see some bias in our estimates. And this is essentially for the reason that I mentioned earlier, that there's not enough information in the mutations to sort of drive the sampler. So we end up having issues both of large confidence intervals, so very broad posterior distributions in our estimates, but also failures of convergence and failures of the Markov chain to mix well. On the right, I'm showing migration results for 2 parameters of interest, the SON divergence time and the effective population size of the African in-group X. And you're seeing results for data simulated with various levels of migration. So as you crank migration up from no migration indicated by a 0 to fairly high levels of migration indicated by a 3, in blue you see that if you ignore that migration in the inference procedure, you obtain fairly strongly biased estimates of the correct parameter values, as you would expect in this setting. When you add migration to the model, so when you do inference with migration in the model, you're essentially able to eliminate that bias and estimate those values with reasonable accuracy. And then the panel on the right is just showing that we can also estimate with reasonable accuracy the overall level of migration that's occurring. Okay, let me jump straight to the results. I might not even have time for the second half of this talk, but I'll jump to my results and then maybe say a few words about the new work that I was hoping to describe. So in the end, we apply these methods to about 37,000 loci across the genome. So these are somewhat arbitrarily selected loci, each 1 kb in length, but selected to avoid all of these sites that are likely to be under selection. So genes, conserved non-coding elements, flanking regions of genes and conserved non-coding elements, and so on, and also selected to avoid areas of the genome likely to be enriched for data quality problems. So low coverage regions, regions with recent transposable elements, and so on. We run the method, as I mentioned, using a variety of different migration bands, and estimate all of the parameters of interest, and then calibrate by making use of a human chimpanzee divergence time of 6.5 million years, as I mentioned. So in this plot, focus on the rightmost bars. These are the estimates with migration, and focus on the, oops, my pointer isn't working. Okay, focus on the rightmost bars. Those are the ones with migration, and then focus on the calibrated estimates on the right-hand side. So those are estimated divergence times in thousands of years. You see that we estimate the African Eurasian divergence time to be about 50,000 years. This actually agrees very well with recent estimates based on a variety of different sources of evidence, including genome-wide genotyping data, resequencing data from multiple loci across the genome, and also with mitochondrial and Y chromosomal data. The SON estimates, however, have not been discussed much in the literature. There are only a handful of estimates out there for the SON. Most of them are based on mitochondrial studies. There were a few based on small numbers of autosomal loci, but they tend to have very, very high confidence intervals. We're able to estimate the SON divergence time much more precisely at just about 130,000 years ago. So it's a strikingly ancient divergence event between the SON, about 2.5 times older than the divergence event between African and Eurasian populations. So it suggests that that these major African population groups diverged from one another long before African and Eurasian groups diverged. I'll say a minute more about that in a minute. We also obtain estimates of population sizes, and I won't say too much about these other than to note that we observe a general increase in population size over time in Africa. So here at the very left, you see our estimate of the ancestral effective population size for the population ancestral to all modern human populations. At the right, you see our estimates for the present-day Yoruban and San populations. So that's consistent with other studies indicating steady population growth within Africa. For the Eurasian, ancestral Eurasian population, we see a pronounced bottlenecks. We see very pronounced evidence for reduced diversity in this ancestral population, reflecting most likely a strong bottleneck event in the migration out of Africa. And this agrees very well with many other recent population genetic studies. The ancestral size, the size of this ancestral population in Africa, we estimated about 9,000, which is sort of interesting, but also quite difficult to interpret because that might have been, this is an effective population size that's glossing over many of the features of that population. So it might have been a substructured population. There may have been variable amounts of gene flow between different subpopulations within this within this larger population, and so on. So this is sort of a very crude characterization of that ancestral effective population, but an estimate of 9,000 agrees reasonably well with estimates that others have obtained recently. Okay, let me move ahead. So, so in summary, it's still striking to me that you can do this kind of analysis with such a small number of genomes. The reason is basically that each of these modern genomes represents many ancestral genomes that have been sort of pasted together by recombination over evolutionary time. What we learn from doing this analysis, however, is that you have to be extremely careful about the way you do your genotype inference in order to get any useful data out of these individual genome sequences. We found that it was important to allow for migration and to deal with the phasing issue with the data. And I didn't say much about this, but we also found it was extremely important to get the methods to a point where they were highly computationally efficient. So we spent a lot of our time sort of tuning the performance of these sampling methods to get them to operate at a genome-wide scale. Our estimates of this SON divergence event at around 130,000 years were in general agreement with previous studies, but somewhat deeper than what had been reported, and with much narrower Bayesian credible intervals, so with higher confidence than what had been reported from autosomal data. As I mentioned, I think it's quite striking how old this divergence event is, 2.5 times as old as the African-Eurasian divergence. It implies these divergence events happened some 80,000 years earlier than the divergence between Eurasian and African populations. But it's important to note that even with this very old event, even with split times on the order of 130,000 years, population divergence times are small relative to ancestral coalescence times. So we're estimating ancestral coalescence times of hundreds of thousands of years with these populations. So human populations are sufficiently large that ancestral coalescence dominates the time since populations have diverged. And that's the reason why we observe more variation within human populations than we observe between human populations, as has been known now for decades. So that observation holds even with the SON. Finally, I should note that this is a complementary approach to much of what's been done with 1,000 genomes data. So the 1,000 genomes data, as some of you might know, are generally low coverage data. The genome-wide data, anyway, is low coverage data. One can do many useful things. The strategy of low coverage sequencing of large numbers of individuals is clearly a good one for variant discovery. So if what you're interested in doing is discovering new SNPs that are not too rare and including them, let's say in GWAS studies, then this low coverage broad sequencing strategy is very effective. But for certain analyses like the one we've done here, it's really nice to have the higher coverage data and to be able to relatively exhaustively characterize all of the variants present in each human individual. So I hope that going forward people will continue to do high coverage sequencing as well as low coverage sequencing, and it's looking like there are several groups quite interested in this complete genomics data as an example. So I think given the time, I think I won't do much with this second part of the talk. I'll just mention that we're working very hard at the moment on characterizing natural selection on transcription factor binding sites, making use of the data from complete genomics, the individual human genome sequence data from complete genomics, and the ENCODE data describing, ENCODE ChIP-seq data describing binding sites across the genome. We're using methods that exploit patterns of polymorphism and divergence. And using these methods, we're able to separate signals from positive and negative selection on particular types of binding sites across the genome. So I'll skip through these slides to my acknowledgments. Maybe I'll give you my summary so that you know the punchline here. So we've developed a new generative probabilistic model for capturing these signals from polymorphism and divergence. It's sort of an integrated phylogenetic and population genetic model. Allows us to use multiple primate outgroups and use them to obtain information about likely ancestral states, which then inform our inferences about selection. The method we're using is designed to avoid dependence on demographic modeling. We can analyze collections of human genomes that might have quite complex demographic histories. So for example, these complete genomics data include several admixed individuals as well as representatives of multiple human populations. We do that by specifically contrasting the patterns of polymorphism and divergence observed within our functional elements of interest. So in particular, the binding sites identified by ChIP-seq with patterns of polymorphism and divergence at flanking neutral sites. And by very very carefully pairing the functional elements with their flanking neutral sites, we're able to obtain a signal that should largely eliminate effects from demography, although we're in the process of demonstrating by simulation that the method is robust to demography. As I mentioned, we can distinguish between positive and negative selection and effectively pool across many binding sites in the genome, each of which has relatively weak information. So many of these binding sites have no polymorphisms and no divergence events. But by looking across a large number, often tens of thousands of binding sites of the same type, we can statistically characterize their average levels of selection. In our preliminary analysis of ChIP-seq data from ENCODE, we do find clear signatures of selection on binding sites, not quite as strong as what's observed in protein coding sequences, but nevertheless quite strong. About 40% of nucleotides on average we estimate to be under selection. And then we find a few transcription factors whose binding sites appear to be under relatively strong positive selection. One of particular interest is FOXP2, and we're carefully following up on our preliminary analysis to try to understand what's going on there. That's a particularly interesting case. All right, so let me just acknowledge people in my group who've contributed to this work. I mentioned Ilan and Leo, Melissa, Armand, Charles, and Brad. Sangchul Choi has been very useful in giving feedback on a lot of this work, and Alon Keenan, a faculty member in my department, has also been a collaborator and has given us feedback on many of these ideas. The a group at Penn State, Webb Miller, Stefan Schuster, and colleagues, were important in getting us initially involved in analyzing the SON genome and gave us access to an early version of that genome sequence data in our initial analyses almost 2 years ago now. On that preliminary data ultimately grew into the demographic study that we've just recently finished. And we've had some very helpful support from a number of people at Complete Genomics in our more recent binding site study, in particular by Aaron Halpern and Steve Lincoln. And so I'll stop there and take any questions.